Here we have the fluid mosaic model of the cellular membrane. We have the phospholipid bilayer. We have the integral and peripheral proteins. We have the sterols. We also have the glycoproteins and the glycolipids. And we also have an extracellular matrix, along with the cytoskeleton. Here we have the phospholipid bilayer. It is made up of phospholipids with hydrophilic heads sticking out and hydrophobic tails facing inward. I will go into further structure on how the phospholipids are composed. So here I'm going to draw a phospholipid. I have the polar hydrophilic head, and then I have the hydrophobic fatty acid tails. These guys will generally move laterally across the cell, and they will rarely flip-flop on the cellular membrane. So how this works is the hydrophobic tail groups will face inwards towards each other to avoid water, while the hydrophilic heads will stick outwards. This arrangement forms the cellular membrane that we're seeing in the previous picture. The phospholipids are composed of long fatty acid tails, a glycerol, a phosphate, and organic compounds. Phospholipids can arrange themselves many ways, but for now we're going to focus on the cellular membrane since that is what we are showing in the big picture. Next, I want to talk about proteins. These can include peripheral proteins, which would be on the outside of the phospholipid bilayer, and integral membranes, which will cross the phospholipid bilayer. It can also be referred to as transmembrane proteins. So next I'm going to talk about integral proteins. So here I drew out alpha helixes and beta barrels. Alpha helixes can be found mostly in eukaryotic cell membranes, whereas the beta barrels will be found in bacterial and mitochondrial membranes. A little bit more background on integral proteins. In order to cross the membrane, you usually have to have 12 hydrophobic amino acids, meaning that their R groups are hydrophobic. And then you usually have 2 on either side of the hydrophilic amino acids, which help anchor that protein into the cell membrane. These guys will move laterally along the cell membrane, and they'll move with the phospholipids. They usually won't flip-flop. So here I'm going over the alpha helix, and this is just a rough drawing of what it would kind of look like if for one single. Usually they can also be found in multiples too. And then the beta barrel, which is the similar situation, it has the hydrophobic center and hydrophilic ends, which keep it anchored in there. This is what the cell uses to get materials into and out of the cell. So I also wanted to mention, just to tie it in with the phospholipid bilayer, the integral proteins, they coincide with this bilayer because the ends, again, are hydrophilic, so they'll be on the hydrophilic end of the bilayer, and the center is hydrophobic, so they'll be able to cross through that hydrophobic section. I also wanted to talk about carrier proteins. These ones can flip across the membrane, but they have to be activated by ions and ATP. So this is an example of what it might look like, and then it would just flip once it has the energy or the ions needed to activate it. Next, I want to talk about peripheral proteins. So peripheral proteins can either be on the inside or the outside of the phospholipid bilayer. If they can either be attached to the phospholipid bilayer itself through covalent bonds or to integral proteins. There's a few different types, which I'll go into further on a piece of paper. So the next type of protein I wanted to talk about were the peripheral proteins. The peripheral proteins are hydrophilic, so they'll either be found on the inside or the outside of the phospholipid bilayer where the hydrophilic heads are found. They can either be bound to the bilayer itself or to the hydrophilic part of integral proteins. So there's definitely a few different types that can be found here. So first we have spectrin. This binds from a protein to the cytoskeleton. So I'm pointing out some examples I have drawn out here. Then we have integrins, which connect the bilayer and proteins to the extracellular matrix. Then we have G proteins, which are just hormone. They deal with the hormone signals that come from the cell, and they can just be found throughout. Since these guys are embedded in the membrane and on the proteins, they generally move laterally with them. They don't really flip-flop across the membrane.
Next we have sterol or cholesterol. They are lipids, which means they are hydrophobic. They will not move except with the phospholipid bilayer. And I will talk about more of their role in membrane fluidity in the next picture. So a little bit more about the sterol or cholesterol's role in fluidity. These guys are embedded within the phospholipid bilayer, so they will move with the phospholipid bilayer and will move laterally. When it's cold, they push apart the bilayer, which helps increase fluidity even though the phospholipids are pushing closer together. And then when it's warmer, they'll do the opposite, and they will push the hydrophobic parts together, which will help keep the membrane intact. So these guys help play a big role in keeping the membrane stable. Another part of the phospholipid bilayer are the glycoproteins and glycolipids. They are hydrophilic and they'll always be on the outside of the bilayer. They play roles in communication and recognition for the cellular membrane, and in organic systems they also help with the immune system. They are covalently bonded to the phospholipid bilayer, so that makes their bond very strong, and they'll move with the phospholipids.